8 kilometers straight down, the water behaves like a slow, cold weight. At this depth, the pressure equals 1,000 atmospheres, the physical equivalent of 1,600 elephants standing in a single column, all balancing directly on your thumbnail. Immense forces at these depths do more than compress hollow spaces. They act directly on the individual molecules that build every living cell. Proteins are delicate, folded shapes, performing every essential function. High pressure forces them to unfold, stopping the chemistry of life. Biochemists calculated this failure reaches a critical limit at 8,200 meters. Past this line, required pressure-stabilizing chemicals would match seawater's salinity. Because this boundary was derived from first principles, the scientific consensus remained absolute. No vertebrate could survive a descent past this depth. The Mariana Trench was first measured in the 1870s by the crew of the HMS Challenger. For over a century, the trench remained written about more than visited, a void where pressure was thought to crush anything with a backbone. In 2017, a Japanese research vessel lowered a baited camera to a depth of 8,178 meters. The footage it sent back challenged the established limits of deep-sea biology. 3 years later, the research ship Pressure Drop pushed further into the Izu-Ogasawara Trench. At 8,336 meters, a single fish glided into the camera's light, breaking the depth record by over 100 meters. This was the Mariana Snailfish. It appeared as a pale, translucent shape with a rounded head and a soft pink glow against the lens. It moved calmly through the deepest darkness in the ocean, showing no signs of distress. By surviving at a depth chemistry textbooks said was impossible, this small animal revised the map of vertebrate life in a single frame of film. An animal this soft faces an environment where physics dictates that proteins should unfold and cell membranes should stiffen. Survival here depends on a molecule called trimethylamine N-oxide, or TMAO. Most marine animals carry small amounts of this molecule to help stabilize their proteins against compressive forces. The snailfish genome contains 5 copies of the gene that produces TMAO, whereas most fish have only 1. This allows for a massive, continuous internal production line. This flood of molecules packs the internal spaces of the snailfish's cells. They wedge themselves into the structures of proteins, forcibly holding them in their correct shapes, even kilometers below the depth where ordinary fish biochemistry fails. The snailfish populates the trench by using these internal chemical stabilizers to maintain the integrity of its cells from the inside out. The snailfish's skeleton follows a completely different logic. A hard, bony skull would tear apart at these depths due to the pressure differentials between internal and external fluids. Instead, a mutation leaves the bones soft and cartilage-like. The incomplete skull has gaps, allowing water to flow through, equalizing pressure. Lacking a gas-filled swim bladder, the fish carries its weight as jelly-like tissue, remaining neutrally buoyant. Its metabolism is similarly adjusted. In coastal fish, the enzymes responsible for muscle contraction slow down under high pressure. In the snailfish, these same enzymes have evolved to actually run faster as the pressure increases. This anatomy allows the snailfish to live as a porous part of the deep ocean, rather than an object resisting it. Life at the bottom of a trench depends on marine snow, particles of decaying matter that fall slowly from the upper ocean. The V-shaped tectonic valleys of the trench act as a funnel, gathering this marine snow into concentrated areas. These zones attract swarms of scavenging, shrimp-like crustaceans called amphipods. The snailfish is the apex predator of this world. It hunts by detecting the small movements of amphipods and using a strong pharyngeal jaw to pull them in with sudden suction. While the abyssal plains above are dominated by slow-growing, long-lived species, the snailfish lives between 5 and 16 years. It reproduces rapidly, thriving in a seismically active environment where silt falls and slumps are common. This dominance is possible because other scavenging fishes, the grenadiers and cusk-eels, hit a biochemical limit around 6,000 meters. They cannot follow the snailfish any deeper. Through millions of years of accumulated molecular changes, this fish has secured an entire ecological tier for itself. It remains the only vertebrate capable of inhabiting the deepest country on the planet.